Hello, everybody. Thank you very much to the organisers for inviting me to talk. For those in the audience who don't know me, I'm Antony. I'm the Director of Neurosciences up here on the Sunshine Coast, and also an Associate Professor at the Griffith University Medical School. I have a long-standing niche and interest in nerve and muscle disorders, so that places me really right at the cutting edge of where things are heading with muscular dystrophies in particular. And I'm really excited to share with you a bit of an update in terms of how research is tracking, including a few locally available trials, as well as what the future is looking like. It's really a very exciting phase for my particular specialty, and for people suffering with muscular dystrophy, the future is looking brighter every year. So the short summary of my talk over the next 25 or so minutes, I'm basically just going to tell you that there is a lot happening. And although it's very much in its infancy still, a lot of the work that I'm going to discuss comes off some pretty hard background work done in spinal muscular atrophy. And that's obviously a separate and very different condition, but it really lets us set the stage for what's going to come. I'm going to comment specifically on trials that are emerging in myotonic dystrophy as well as facioscapular humeral dystrophy, because both of those have locally available trials that are currently recruiting. And we'll talk a little bit about future directions and how to get in touch regarding any particular trials. So, a lot of the information that I'm going to discuss over the next little while just need a little bit of background knowledge about genetics and how genetic disorders such as muscular dystrophy are generated. So for most of us, we have all of our genetic material organised in 23 pairs of chromosomes. On these chromosomes, we have a large number of genes. So the human genome has about 20,000 different genes. Genes are broken up into structures that code proteins, and proteins are the building blocks of life and function. In a gene, it's made up of exons and introns. Exons are the bit that actually contribute to the code that generates the protein, and introns are bits of the genome that are not explicitly expressed, but they probably do still have critical functions. And when our body reads the genetic information to translate this into RNA, which then can translate further into proteins and amino acids, this DNA or the genetic material needs to be spliced into RNA. So all of the exons are pulled out and the introns are left behind, and then the exons are joined together. And then this RNA is translated directly into the proteins and amino acids. Most neuromuscular diseases relate to one of two major problems. And one of those is that a mutation occurs that results in a particular gene losing function. So the code gets jumbled and the output becomes nonsense. And so a protein that's necessary for some cellular function is not able to be produced, or a gain of function, in which case a superfluous or toxic protein is produced excessively, and that causes deleterious effects on the cell. Now, spinal muscular atrophy is kind of the poster child for what can be done successfully with modern genetic technology because, well, for several reasons. One is that it's a terrible disease. So there are broadly 3 or 4 different types of spinal muscular atrophy, most of which present very early in childhood. Spinal muscular atrophy is mediated by a gene called survival motor neuron 1 and 2, and about 1 in 50 of the healthy population have the loss of one of their 2 copies of survival motor neuron 1. If 2 people, each with a deletion of this SMN1 gene, get together, there is a risk that one of their offspring could have a deletion of both the survival motor neuron 1 genes, which means they're unable to produce this particular protein. And the severity of their disease then relates to how much of the second gene, SMN2, they have on board. And you can see in this diagram over here on the right, a normal healthy 5q chromosome has both SMN1 and SMN2. Type 1 spinal muscular atrophy, which typically comes on in the first few months of life, has no copy of SMN1, but has 2 healthy copies of SMN2, which are then able to produce some survival motor neuron protein. SMA type 2, which comes on a little bit later in childhood, will have often 1 preserved copy of SMN1 and at least a couple of copies of SMN2. And then the milder phenotype of SMN type 3 and 4, which come on in adulthood, typically have a single copy of SMN1 and numerous copies of SMN2. Type 0, which is the most severe form of spinal muscular atrophy, has none of the above, and that's a very rapidly fatal disease, unfortunately. So the life expectancy of these is less than a month for type 0, 6 to 18 months for type 1, about 2 years for type 2, and sometime in early to mid-adulthood for type 3. So devastating disease, affects children very, very severely, and is essentially a type of pediatric motor neuron disease. And a lot of this relates to how SMN2 functions. So SMN2 has a particular exon, number 7, which is essentially randomly included or not in the output. And in the average healthy person, only about 10 or 20% of exon 7 is included in the output, which produces normal, healthy survival motor neuron protein. And the other 80% of the translated protein lacks that. So it begs the obvious question, what would happen if we could encourage the body to include exon 7 more? That's essentially what we now have the technology to do. And this was developed first by a technique called an antisense oligonucleotide, a drug called nusinersen or Spinraza. And what this drug does is, as you can see down the very bottom here in panel C, nusinersen encourages exon 7 to be included in the transcript, and it does that by blocking a specific splicing silencer, which means more of the RNA that's produced from the DNA includes exon 7, and thus more of that exon 7 containing RNA can then go on to produce the normal survival motor neuron protein. This particular molecule is very large and can't cross the blood-brain barrier, which protects our brain from all sorts of exotic substances. But the biggest downside is that means it needs to be given into the spinal fluid. So a technique called intrathecal dosing, where a lumbar puncture introduces a fine needle into the, usually the cerebrospinal fluid in the lower spine. And this drug is injected every 2 weeks for 3 doses. A 4th dose is given a month later, and then it's 4 monthly dosing thereafter. So that worked well, and the data was really encouraging in terms of improving kids' health and stopping them from declining, but it's a stopgap. You know, every 4 months, injection into the spine is not fun for anybody, let alone small children. So the next technology that came along was the recognition that viruses can be genetically modified to reduce their pathogenicity, so reduce their downside. And because viruses are quite aggressive, invasive molecules, they can be custom designed to help us introduce new genetic material. And if this sounds like something very space age and science fiction, that's because it is. This is an incredible technology, and for lots of different reasons, it's delightful that this has worked as well as it has without significant problems. But onasemnogene abeparvovec, or Zolgensma, has been used basically using a fragment of an adeno-associated virus, number 9, to introduce survival motor neuron 1 into affected motor neurons. And the really amazing thing about this kind of technology is it's a single infusion. So a simple infusion into a vein once in your life, and the abnormal genetic makeup that has caused the disease is not quite, but essentially fixed. And what this therapy means is that if you can make the diagnosis super early and give someone this kind of therapy before they develop significant symptoms or significant disease, you may prevent their subsequent decline. And that's exactly what this has done. So it's now possible to test embryos in utero and give this therapy essentially immediately, and that child will then have almost normal motor function. So huge life-changing drug, incredibly expensive, as you might expect, but nonetheless groundbreaking and very exciting. And then the third therapy that came along in spinal muscular atrophy is called risdiplam, or Evrysdi. This is a small molecule that modifies splicing, and because it's such a tiny molecule, it's able to cross the blood-brain barrier. And that means, for the first time, this therapy can be given orally. It works in much the same way as the antisense oligonucleotide does. So it increases exon 7 inclusion in the RNA coding, and so it increases the survival motor neuron protein production. And this is now readily available throughout Australia and essentially can help people, instead of getting 4 monthly lumbar punctures and fairly uncomfortable procedures, this is a simple tablet that people can take. And so all these three therapies really laid the stage. That's spinal muscular atrophy, but could we do the same thing in other diseases? And the short summary is absolutely. And so the first off the rank is myotonic dystrophy. Many of you in the audience will be familiar with this disease, so I won't rehash it too much, but it's one of the more common muscular dystrophies we see, about 1 in 2,000 people affected across the world. It relates to an abnormal repeat of 3 particular elements of the genetic code at the end of a gene called DMPK, and the number of repeats of this code correlates fairly closely with how bad the phenotype is. And so more than 100 repeats, and the person will generally have classic myotonic dystrophy. A lower number of repeats heading back towards the normal range can give you very subtle or sort of pre-mutation status. And most unfortunately, myotonic dystrophy with many repeats is a fairly unstable mutation. So each subsequent generation tends to develop more mutations and more repeats than the previous generation, which means your children tend to be worse off than you are. And the mechanism of this is because you have this very long region of repeated genetic material, it causes dysregulation of the normal splicing of the RNA, and ends up with a mutated DMPK protein with an unusual hairpin loop structure, and that traps another protein called muscle blind-like protein, or MBNL, and that particular protein has a really critical role in splicing regulation in skeletal and cardiac muscle, as well as the function of chloride channels. And chloride channels are the reason myotonic dystrophy has myotonia as a symptom, so impaired muscle relaxation. So myotonic dystrophy in the last 5 years has really seen an explosion of work. And if you look at clinicaltrials.gov just yesterday, my days are going to be out of date by the time you see this recording, but within the last few days, there were 126 active interventional studies looking at myotonic dystrophy, some of which have completed recruitment, but many of which are in the startup phase. And the first of these I've highlighted there, nice paper in the Lancet from not too long ago showing a very early trial of an antisense oligonucleotide was safe and well tolerated. And there's early signs of efficacy with these therapies. And currently in the area, these are the 6 trials that I'm loosely aware of, all at varying stages of startup. And I'll run through them very briefly, one by one, just to give you a bit of an overview for what these different trials are looking at. But they're all going down the very same path that spinal muscular atrophy did. So proper, potentially disease-modifying therapies are coming. ARROW-DM1 is the first trial to mention because, very selfishly, we're one of the sites recruiting for this study. This is a intravenous RNA interference molecule that's job is to silence the abnormal DMPK protein within the muscle. Arrowhead Pharmaceuticals are the sponsor of this trial, and these are the inclusion and the exclusion criteria. The inclusion criteria are fairly broad, and the exclusion criteria can be challenging in myotonic dystrophy, especially around clinically significant cardiac disease, because it's relatively common to have some degree of cardiac conduction issue with myotonic dystrophy, even without any cardiac problems. And so recruiting to this trial and finding people who are able to participate can be challenging. The trial itself is relatively short, so up to a year for the phase 2A arm, but only 3 months for the phase 1 cohort. It does involve some needle muscle biopsies, which is a turn-off for some people, but has been generally pretty well tolerated. And that allows the trial to assess for different muscle markers through the trial, including the level of the DMPK protein, which is obviously critical in terms of making sure this drug does what everybody hopes it will do. There are a decent number of study visits and a reasonable number of participants. So phase 1 is looking at 12 participants to make sure this type of therapy is safe and well tolerated. And then the phase 2A with a larger number of participants will hopefully be judging whether or not there's signs of efficacy and pursue a larger phase 3 trial. Information is relatively easy to access through a centralised email. So if you or people you know have myotonic dystrophy and are interested in finding out more about this trial, please get in touch with Arrowhead. There are 3 sites in Australia, us on the Sunshine Coast, Rob Henderson in Brisbane, and then the Alfred in Melbourne. So most people on the eastern seaboard are catered for, and obviously we're more than happy to discuss things in detail if there's any interest. Dyne Therapeutics have a reasonably exciting molecule under phase 1 and 2 development as well. This study, I believe, has completed recruitment or is very close to completing recruitment, but has not yet fully reported its data. There have been some sort of early press releases about the safety of the molecule, and they have reported some improvements to clinical markers such as hand opening time. So myotonia, people will know if you make a fist, your hand will tend to get stuck and then only very slowly release, whereas this molecule has helped people get less myotonia. They've tested muscle strength as well as some functional assessments, including how long it takes to walk or run 10 metres and how easy it is to get up and down from a chair 5 times. And all of those markers seem to be trending towards improvement. This is a therapy also that's given intravenously, so it's really nice that in this field people have taken note of the difficulties with intrathecal therapy seen in spinal muscular atrophy, and there's a real push to get well-tolerated, much more easily delivered therapies, and that's quite exciting to me. Dyne's molecule is an antisense oligonucleotide, so very similar to nusinersen, except it's specifically bound to a special fragment antibody that binds to a muscle receptor called transferrin receptor 1. So it's facilitated entry to the muscle that way. And because this molecule is targeting the muscle directly, it means we don't need to worry about the blood-brain barrier and those kind of delivery concerns that need intrathecal access. Freedom 2 DM1 is another phase 2 trial that is just in the startup phase, I believe. I'm not aware of it currently recruiting at the moment. A drug company called PepGen is behind this one, and it uses antisense oligonucleotide that binds to the abnormal repeat expansion in DMPK RNA. The plug that the company have given this molecule is that they believe they're targeting only the abnormal DMPK, the one with the abnormal hair loop structure. Theoretically, this will leave normal DMPK behind. And I guess it's important at this point that the function of DMPK is not perfectly clear. So a lot of these things are very complex and we're really only scratching the surface in terms of knowledge. But there is, if you have the ability to design a technology to target only the abnormal RNA and leave normal RNA behind, academically, you can understand why that seems like a tantalizing path that may give some advantage that we're yet to measure. This also is easily delivered via an intravenous infusion, and they're looking at a placebo-controlled trial to look for early signs of efficacy over 3 months or so. The BRAVE study is very early and is yet to start up in Australia, but they are looking for sites and soon to be looking for participants, I believe. So this is led by Sanofi, which is a fairly large pharmaceutical company. They have replicated what anasemnogene did in spinal muscular atrophy, and they've used an adeno-associated virus number 9 based molecule, and this also targets DMPK RNA. Just like the amazing work in spinal muscular atrophy, this will be a single intravenous infusion. Obviously, being a phase 1 study, they're not looking for proof of efficacy at this stage. It's more checking that the molecule is safe and doesn't have any unintended side effects. But potentially, this will be a therapy that particularly families with myotonic dystrophy may be able to get pre-symptomatic testing in childhood, work out who's at risk of developing myotonic dystrophy, and then have a therapy like this to prevent them developing myotonic dystrophy. So amazing to think that within my lifetime, the degree of work I have to do clinically in my clinics may be very selfishly cut down, which I think is just fantastic for the community. VX670, most of these drug names become a bit more catchy once they've proven they work. They don't waste time on the marketing until they're shown to be effective, so they can be a bit hard to keep track of. This molecule, though, is developed by Vertex Pharmaceuticals and is also, again, another antisense oligonucleotide infused via intravenous routes. This trial is underway, I believe. We're not involved on the Sunshine Coast, but sites at the Wesley in Brisbane and the Alfred in Melbourne, I'm sure would welcome contact if people have any interest in looking at this. And you can see there the inclusion criteria are fairly broad, so you need to have a genetically confirmed clinical diagnosis of myotonic dystrophy, and then there are some exclusion criteria that are not available on the clinical trials website, but would be available to the sites to go through screening questions with people. And then lastly, del-desiran is the latest phase trial on this list. So a phase 3 trial, which is really towards the pointy end where they're trying to show that this is in fact an effective therapy and can be used clinically. This is being developed by Avidity Biosciences, and you can see they're getting close to working out it's a good thing because they've given it a proper name. It used to be called AOC 1001. This is a small interfering RNA molecule, so very similar to the risdiplam for spinal muscular atrophy. It's conjugated with a transferrin receptor 1 antibody, which is that same one that some of the oligonucleotides were targeting to get access to muscle, and targets the DMPK RNA just like the other molecules. Although this is a small interfering RNA molecule similar to risdiplam, it's still given as an intravenous infusion. And I confess I don't understand why they can't develop this into an oral molecule like the risdiplam, but I'm sure there's good reason. It acts basically in much the same way as these other agents. So all of these molecules are targeting the same pathway, just taking slightly different approaches to the same problem. But makes me very optimistic that they feel they're on the right track when so many groups are doing the same thing. Moving on to the second muscular dystrophy that I thought we might cover today, and that's facioscapulohumeral dystrophy. Many people in the audience also will be familiar with this and know that it relates to sort of the opposite problem of myotonic dystrophy. So the normal facioscapulohumeral gene, just distal to the DUX4 gene, has usually 11 or more D4Z4 repeats, and this results in normally inactivation of the DUX4 gene. So outside of embryogenesis, DUX4 is not expressed in humans. When facioscapulohumeral dystrophy occurs, there is a reduced number of these D4Z4 repeats, which means that DUX4 is inappropriately expressed, and that causes the downstream effects of the muscle disease. You can see the Latin roots of the names in the name of the condition imply involvement. So fascio means face, scapulo talks about the shoulder blade, and humeral usually refers to the upper arm where the humerus is, and that's because biceps is often preferentially weak. The clinical features are just like the picture on the right there, so scapular winging, facial weakness, biceps more than triceps weakness, and sometimes distal leg weakness, particularly around ankle dorsiflexion. There are 3 relatively exciting trials in this area, all of which are very similar to what we've just been discussing, and all of which are in fairly similar phase of startups. ARRODUX4 I'll mention first because, again, selfishly, I have a bit of an interest here in that we're running this locally as well as other places around Australia. This is, again, you can tell by the naming, it's Arrowhead Pharmaceuticals again behind this one. Inclusion criteria are fairly broad and the exclusion criteria are listed there, which basically revolve around other health problems that may impact an ability to conduct a trial. Again, very similar to ARRODM, this study is looking at how well the molecule works from a safety perspective more than efficacy. And it involves urine and blood samples, as well as at least 2 needle muscle biopsies, one pre-dosing and then one after dosing. It's an intravenous infusion, this product, and the muscle biopsy is guided by muscle MRI. So they're, in this case, using centralized reading of a whole body MRI. So for people who are highly claustrophobic and can't do MRI, this may not be the trial for them. I was our local site's healthy subject for muscle MRI, and I have to say I was in there for a very long time getting all the protocols right. And so I can definitely sympathise with people who don't enjoy MRIs. There is only the one MRI, however. Once it's done to target the first biopsy, the second biopsy is done of the same muscle at day 42 without further MRI. So it's only the one that people need to get through. This trial is looking to recruit around 24 participants with FSHD and work out how this drug behaves at various different doses and what effects it has on the muscle parameters that they can measure. More information is available at any of these 4 sites within Australia. So locally, these are our generic contact details for clinical trials, but the Wesley Hospital in Brisbane, Liverpool in Sydney, and the Alfred in Melbourne are also involved in this trial. People nearby to them can reach out for extra information. EpiCRISPR is a really exciting trial that is not yet active, but has a really interesting mechanism. So it's using a different adeno-associated virus, rhesus 74 is a particular adeno-associated virus vector that they're using. And they're delivering a CRISPR-associated protein to bind to D4Z4 and then directly repress the expression of DUX4. So, if our experience in previous trials and other diseases is anything to go on, there's good reason to believe that this therapy could be a good thing, but of course it's early days and really needs to go through the whole trials process to assess. At this stage, the only 2 sites that are listed on clinical trials are in Georgia in the US and Auckland in New Zealand.